you want it to be. Okay. So that's what you should all take. And then we're not gonna say anything. This is just a demo in kind of product design or trying to understand educational context. So everything you need to do is in that white pouch. So try to just open it up and get started and see what you can figure out by yourself.
How different is Foldscope 1.0 versus 2.0?
It's a massive difference, and I will ask maybe Kevin to bring. Can you bring 1.0 from downstairs? It's in the same box. Where there were boxes, these very colorful plastic bags. Yeah, yeah. Can you just bring one? I just want to show design contrast.
I'm not normally very good at following directions.
You'll figure it out. Just everybody should follow directions and see. Imagine you're a 10-year-old in the middle of nowhere. You somehow magically this pouch landed in your hand.
Oh wait, this is way different than the 1.0.
It's completely different. It's like totally a different object. But see if you want to follow instructions, do what you want. But if you want to follow instructions, everything is in that little paper booklet. And it's color coded, and you really do want to look at the colors carefully. So, and then there is no glue, nothing, so you can assemble something and it will suddenly, you can go back, it's not a race at all. Imagine you're in your room late at night, nobody's watching you. Take your time, play around.
Did you say follow this instructions or did I not?
No,
I say,
okay. Sorry, I didn't mean that. I meant I am not gonna tell you anything about what to do with the object, but the object includes a little booklet. So it's not a quiz, it's not a test of anybody's origami talents, nothing. You can talk to someone who's around you. I just don't want to bias your experience. I want you to feel as if you were a kid that just got this object and your teacher walked away because they wanted a break. And what happens next? It's not fragile, it won't break. It has magnets, so you can figure out that you can open and close things. We're just following instructions, whatever is in that paper. But you're also thinking in your head, and again, of course, we train the trainers around the world for this, but I'm just doing the quick experiment. And then if you figure out a step, what you're trying to model is that look right and look left, and you can help your neighbor who's figured out that step, because it's not a race. You want to just be able to share knowledge. And it's color coded, so colors matter. So,
Go back and forth.
But
you go forward,
yeah, like that. The subtle point.
But I think you need to.
Yeah, help support each other. They will tell you what you're doing if you just sit next to it. So somebody can just mention what we're doing.
Yeah.
Except line up Foldscope using the instructions that are in the white.
Hello.
And I think this part, it's just.
Only steps makes it easier. Oh, he's gotta go up through here. This is kind of. Open that up and then fold on the corners.
Yeah.
So I can fit.
What is this? How to get it? Yeah, if you send me your address, well, we will send Foldscopes to your address.
Yeah, this needs to go like a layer behind it. This is behind them.
Oh, so,
the Foldscope? Okay.
yeah, I think everybody is, so just have,
Can we get a spare?
I think it looks like, I think.
Go.
No. I think somebody's in that right now. Yours looks a lot better than mine. Flip it over. Some folder. Oh, I think this.
It's backward.
Yeah.
I think it's just the insert.
Oh, this guy.
So open it up. Just mag. And then you gotta split that. How many prototypes did it take to get to this?
this for 10 years. So you can imagine, evolution of a design for 10 years. Say officially, this is like 350th
Yes,
version.
Back to
But we just call it Foldscope 2.0. We don't release all the designs,
like this is.
but there is and also
There you
these designs have
Yeah,
spawned
yeah.
out. And there are many versions and variations. Now there are polarization instruments, there are fluorescence, Foldscopes, there are dark field instruments, so there's a lot that not a single, not even a single. And if you are assembling, then tell your neighbor sets of steps. They have commands. Some of you are almost done. So maybe I'll just say a few words sort of philosophically first. So hi, everyone. Sorry you don't have, how many of you have this object in hand, by the way? Because I know some of you do. Yes, so I'm hearing a few sets of names, but one of the points around this is we're going to talk about paper today, and just I want to start with design philosophy of how this object came to be, and then talk a little bit about how to build solutions in this context. But before that, it's important for you to kind of see the sets of differences. And then, of course, before any of the philosophy or what are we trying to do, the purpose of this is to experience the microscopic world. And so you've assembled something, and I might say that, oh, what you assembled is a microscope. And you don't have to believe me. It's just a piece of paper. Many of you have been assembling, so just quick sets of instructions on assembly, and then we'll talk about design for why it looked like that, now it looks like this, and there are many other variations. But just an important thing is to now, if you hold it in your hands, it should look green on these two sides, dark green here. When you open it, there should be this little pocket that's open. And if that's not the case, then unfold it and try to see. So there should be these two little arrows. Do you see that?
Mhm.
So it should exactly look like this because everybody has the exact same part. So for example, that one, I'll pass that for a second, and it doesn't look like that. And there's just an orientation factor. And so what I'm just going to do is retrace our steps for a second. And everything is actually perfect, except just the one step, which is just an orientation. That's it. Because you want this accessible, and you can play around with it in terms of thinking about and everything else was exactly the same. And so now you will have the little knob and it'll immediate. So some of you have lenses you've already mounted and some have not. So open the pouch and there's a magnetic lens inside it and it just snaps right here. So there it is. And then let me take a look at this one for a second. It's the same. Yeah. So you want to just flip that because you want that in your hand. And that's for the phone. So when that thing comes, it's attached to the phone, you peel that off. Yeah, that's the lens. And then because it's a lens, you don't want to put your thumbprint on it. That's sort of obvious why. But let's actually transition to see whether this is even a microscope or not, because you've built something. It's like having a new bike that somebody tells you is a new bike, but you don't know how to ride it. You don't know how to ride it, it's almost worthless. But if you know how to use a bike, then you can go places. Okay, so let's try going places. I'm gonna borrow one Foldscope, for example. So everybody should be here because now we're gonna kind of accelerate. Right? Everybody has that. Don't worry about using it yet. So now I'm gonna show you how to use it. Yes, but everybody is here. I'm gonna get that show of Foldscope. Yeah, take your time. Take your time. Yeah. Yes. Almost there. Yes. Okay. So let's actually wait for everybody to get there. So we will okay, so while we're doing that, this object has evolved dramatically. Many years ago, it used to look like this. Exact same function, totally different design, completely different folds, but exactly not a single feature we didn't want to lose. This takes roughly around 25 minutes to put together, which some of you have. This, I think I didn't tell you anything, but I would say, 5 minutes was a good estimate of thinking about it. It was a huge design choice for us to make sure that with zero instructions anywhere in the world, just with pictures. There's a lot of, this is a particular print. There are other prints that we do on top that are really steps are on the print itself. So this is also media. You can actually print on it. Instructions are color coded stepwise. So you say, oh, start with the rainbow color. So we do all kinds of funky things depending on things that we're playing around. But there's an important aspect of evolution. And because that came from the pressure where the teachers were like, we only have 1 hour with the kids, we don't want to spend the first 20 minutes only making it. We really want to actually build stuff and play with them and walk around. This is an object, if I tell you, yes, it's actually exactly the same price. These are out in the world now. There is 3 million or so of them out there. Every one of these objects, this is roughly around, I think maybe currently, if I do the numbers right, this is like $2.75 or something. This used to be roughly around $1.75. There was a version before that that was around $1.50. And kind of the structure and evolution that has happened is the capabilities have grown. So there is a separate kit to the side that I'll talk about. Some other design choices we made was we made swappable lenses. So now there are 3 objectives. They're different, and I have some of the other objectives here. I'll show you guys. That allows for anybody to manufacture an objective, but then also it gives them the notion if you have these other objectives, you can start looking at different magnifications to begin with. This used to only have a single objective. That kind of was the big technical jump to make. To go from there to there, everything had to change, like from scratch, except we want the experience to be the same. Okay, so I have said that it's a microscope, but we have to prove that. Something that's new and playful. So this is backward compatible technology. So the first thing we can do is I'll just pull out a glass slide. And this is not the experience of microscopy I want to have for people, because this is a pre-made object. But I just want to show it to you that was another design choice, because we wanted to make sure that people's perception of microscopes comes from these glass slides. In teaching and education, all kinds of slides are made and used. So this object, which you're all very familiar, how many of you do microscopy regularly? So everybody has used the glass slide, right? So that opens up, and at the back, you see this pocket. Now this has a cover slip. So the cover slip goes towards the objective, and you tuck it in this direction, so that it's completely inside the pocket, right? So now at this point, I'm holding with two hands, always hold with two hands. This is like when you drive a bicycle, put your hands right there. The thumbs are literally printed on it. This is X and Y. So you're going up, you're going down, sideways. Z is your focus. And you can actually see the design. It's stacked thicker on one end, thinner on the other. There are magnets that are pulling. So it's a magnetic flexure. Remember what we talked about compliant mechanisms yesterday. If you notice it, there are two thin points in this. And so there is a pivot point, and that goes up and down. So this is tens of microns of very precise movement. But it's not cantilevered. It has two cantilevers on the two sides. So it remains balanced. It's not hanging. So it doesn't vibrate when you don't want to. And so this little sets of tricks. There is a magnet on the other end. We tune the strength of the magnet to make sure this can actually slide. If this was a little stronger, it won't. So now at this point, I'm just using ambient light, and I'm actually doing microscopy, and I am looking at a sample. And one of the key things around this is what I'm going to do is we're going to spend some time now making your own sets of slides, which is very important. And if some people don't believe me, I'll pass this around. So now you hold that and then just point to the light, and you should see single cells. You should see nuclei stained.
Wow.
Do you?
Yeah.
We also built a detector. It was a very secret technology. If the person is not smiling, then it's not working. So I mean, I didn't pay Danny today to do that. So pass that around. Take a look at that one, Peter, first. Yeah. So, I think it's kind of a point about when you build something, it should itself just share the expression that you don't have to be there to convince people. That's a very important aspect. But this is just to prove that the microscope works, that it is actually a microscope. Now we're going to get into and one of the things that's really fun about experience is it's honest. It's sort of, you can never take it away. So you want to use two hands, and then you can move around that way, and you have XY control, and then you want to make the focus to be really, really sharp. Okay, so while this is being passed around, we'll also show that to the class in a second. And make sure, unless it's working incredibly well for you, don't pass it to the next person. Just play with it to make sure that, yes, it actually works. Okay, so now if you've experienced that, now we flip this thing around. That was a pre-made object by somebody. That's somebody else's idea. Like, you have to start with your own idea, because all of exploration and observation is about your own observation. So in this recent one, we've released these little playing cards, and we're going to play a card game now. So this is a little piece of paper. You can make as many of them. So it looks and feels like a trading card, and it is a trading card. Now the question is, what do you guys want to look at? And if you claim it, nobody around in the room can claim it. So tell me, what do you want to look at? Look around you. Yeah.
Hair.
Okay, that's done. Okay, let's keep going. The more wilder the object, we could actually walk out for 10 seconds. Why don't you guys?
Yeah.
You have, the clock starts now. You have 5 minutes. Let's see if you bring the wildest object from your imagination. I am starting the clock. So everybody, actually now I can spend time with you guys while these guys are, my stopwatch starts now. 5 minutes. Okay. Bring the wildest object you think. Whatever you think you want to bring. Something that's going to surprise. You can go outside, you can be anywhere, but 5 minutes only. Okay, sorry, I am just letting them all run. One thing I will do is go back to, Kevin, can you show me how to do that view again, the gallery? I like this way. Okay, perfect. Can people hear me? Yes? So actually, while people are searching for wild objects, oh, Kevin, you can give them these. They forgot to bring. No. Ziplocs. Why don't you take a few of these and are around? This has Ziplocs, little Eppendorf tubes in case people want to bring sample. At least bring me something liquid. Actually, can you bring me spirostomum from downstairs? Our culture. Yeah, they have Eppendorf tubes in them. Okay, while they all went to look for things, I'll just talk a little bit about paper. And I'm assuming if you have your Foldscope, you can go search for an object itself. Any questions so far in what I talked about the design of this object, any of the technical sets of threads? And I'm assuming some of you have this object, but some don't, right?
Professor, oh sorry, Manu.
Yes, go ahead.
You talked about compliance design. So yesterday when you were talking about the small sharpener thing, is this the
Yes.
one related to the Foldscope? The X, Y, X axis, Y axis in the right one. Somehow is it related?
No, that's totally different project. Sorry. Yeah. Foldscope is completely made out of paper and magnets and lenses, while what I was telling you was something kind of on manufacturing. I see Manas's hand and Debajyoti. Manas, do you want to go ahead? And then Manas, can you introduce yourself as well, just briefly?
Hello, Professor. I'm Manas Rawalpalli. I'm joining from Ashoka University. I just wanted to ask, what was the use of the magnets that you mentioned in the Foldscope?
Yeah, so there are a couple places magnets are used. So first of all, the lenses have tiny little magnets on the back. And so then they snap. So optical alignment of this to the other aperture. There's another aperture on the other side. This aperture gives light. So we want to make sure these things are aligned. And if you guys want to, I forgot to tell you, all of you have these little Eppendorf tubes and things so people can get something. So that's the magnets. The other part is that there's a magnet in this as well, right there. And this generates a tension. It's attracting the lens. And so now the lens distance is fixed. It's very important for a microscope objective to have, so what we call this is a magnetic flexure. The restraint is provided by paper and the attractive force is provided by the magnet, and the balance of the two gives you a stable displacement. And why is it a flexure? Because if you notice on this side, it's thick here and thin here, and there are these two thin ends. And these thin ends allow the lens to move single micron digits when this is being moved in such a floppy object. So although this is floppy, the actual optical assembly is quite stiff. And to provide that stiffness, we need magnets.
Okay,
and it's also counter forces. Okay, I'm going to do a few more questions in a second. People have come back with samples, so I just want to tell them what to do. Okay, so wait a second. Don't put anything yet. You have to now learn how to make a sample. So take your trading card. Did anybody bring a wet sample? Awesome. Okay, so the first thing you want to do is take out this little plastic film, and it has a little piece of tape through your nail, and you put it on one side, even without putting the sample. And so now suddenly you see we have created a little chamber. If it's a dry sample, you can just mount directly on it. But if it's a wet sample, we're going to put that little drop here. And I have absolutely no idea. And don't tell me yet what it is. Okay. We'll just, we'll figure out, because remember, we're playing a game. And then because it's a wet sample, I'm going to take a second little piece of tape here, right there, right? And put it on top. And that should seal it. So at this point, you can see, I'll pass this around. So this is an example of how a wet sample should be mounted. And you see it's completely sealed and has just the sample in it. So you could have put pond water, you could have put whatever. Now, some other samples might be too thick. So that's an important thing to think about. There are tweezers in there. So when you want to mount, for example, let's choose this flower.
I was gonna take off the pedal, but.
Yeah, no, but what I'm just saying, any example for a second, if you're gonna take the pedal, and if you feel it's too thick, just tear it. So when you tear it, it generates these thin layers, and some of the single cell layers will then be visible. So in some sense, play around, and as you play around, mount this. So just the final bit on this is this card goes exactly the same way. So if you all just look here for 1 second, see the two knobs, it's very important that it goes in the pockets, because if it doesn't go in the pocket, you will not have the XY resolution, the movement. So it needs to fit, right? So it needs to fit right there. I mounted it and the guesswork begins. So I'm gonna pass, this was Enrico's, and sign your card, because this is a trading card game. Don't write what it has, just sign it, and other people are gonna figure out what you've just mounted. And if you figure out, you get to steal that card from the other person. I should have told you that. So let's see who collects the most cards. And then we will have a battle.
And then how do you?
Okay, this is
and
gonna
then
be
you probably put another
hard.
one
Yes.
on top of it.
Ooh,
Secret
that's
sample.
secret sample. Okay, I'm gonna pass this in a second. Oh, this is super, super cool.
Yeah. Oh, yeah. Only just maybe.
Okay, I'm gonna pass this secret sample around if anybody wants to look at Enrico's sample. And I'm gonna mount another secret sample. And then if you have it, you've seen it, pass it around to your friends to let them start guessing. So I'm curious if you, did you make it already? Sample one? Oh, your Foldscope I was using as a demo. Enrico, where was your
It
Foldscope?
was this one.
Okay.
Maybe.
What did you do with your Foldscope? I literally had it. Num, num, num. I'll just build one for you. Yeah. I'll put it together in a second while we, oh, you have the sample mounted? Can I see what it looks like? Yeah, you want to use
No, I think I
the
can
one
see it.
first. So like the trick could be is, so one is we can just first pick it up. It flew here. Bad idea. There it is. There it is. So because it's charged, you see what's going on? Yes, which is super cool. Now you can start asking why would one be charged?
On the one backside.
If it's a dry sample, it's perfectly okay to just have one single side. So this is good. And then I'm gonna assemble this for 10 seconds. Effectively 10 seconds.
I think I'm going to my object at the back.
Yeah, I'm gonna teach you guys how to mount lights in a second. First of all, try the indoor
Right,
light,
yeah.
just try the outdoor light. So before you get to the
So
flashlight,
what I usually do is
it's
I-
valuable to just come on the window, because
Yeah,
you'll see, so just, you can literally walk to the window.
You
The
like,
closer you are, the better it is
Oh,
in
that's
terms
the
of
thing.
artificial or real light.
Oh,
So that's done.
that's crazy.
And then if you have sampled something,
And you can turn it.
you can start drawing and get a sense for what you are looking at. And then also share that with other people so that they get a sense for what they're looking at. Okay, so we have a unit. And I don't know if people have already figured out what other folks have mounted. Okay, so we mounted that, we have it here. And then usually I like putting it closer to the lens.
Maybe to like. Yeah, I don't know. Maybe it's like, or like the cat. Yeah, I don't know. Yeah, like, yeah.
I am just to make sure.
All right, who wants to take a look at this guy?
Your hand.
Right there.
No.
See it so that I don't have to keep searching. Close it. Bring here, I always bring
That's
this
so
to
cool,
0.
right?
So just as quick, whenever I put the sample, I first bring it to 0, because then I know that the focus is only on one direction. Just kind of helps me. And already,
No, it's not. I wanted to do here, but
okay, take a look. This is pretty spectacular, actually. Both here and here. Okay, so draw some pictures, and then those pictures you can doodle and draw on the card itself. And then can we share a few sets of ones? I want to see. And let's see if we can show the class for a minute.
nice to see you. This is really cool.
So don't open. Ooh, that is super cool. You're not allowed to open when we are trading. So I know what this is. So I can steal this card. But it would be cheating if you open and look at it macroscopically. Now, I'm not going to steal it, but this is spectacular, I have to say. Okay, let's pass around. So trade your Foldscopes, and I'm going to show the class actually for a second.
Wait, and we guess if we get it right, or
If you guess it right, then the person that you are trading with, then you can collegially request that you win the card. But you have to be able to say quite a lot about the object. You can't just say, oh, it's an insect. That's not what it is. So you have to say something. You have to understand the context, what just happened. Did you guys trade? Did you get a chance to look? Can I see?
wait, move it around because it's more than.
Oh, and then make sure you
wait,
mark.
that's not mine. That's not.
Okay.
No, no.
Ooh,
which one?
that's beautiful.
Yeah, but I want to wait.
And
Yeah.
actually, it's quite dark in the room. If you guys come to the window.
Well, this one's not mine. Are you okay? Mine.
That's the one you handed.
No, the one I handed you is mine.
Yeah, you can find it. By the way, you can also write an initial. So play around, get a sense.
No.
The goal is to really stump the other person first. Oh, it's spectacular. Who's whoever this is? The purple?
Wait, purple's good.
Oh.
Purple's good.
Okay, but you have to be able to say quite a lot. I'll let you guys all talk.
Wait. Hold on.
What you got?
Okay, let me ask you a quiz question. Especially to the plant biologists here. Do flower petals have stomata?
Does depend on the particular petal.
Flowers. What do other people think? Do flower petals, I mean, we know leaves have stomata, but do flower petals have stomata? Have you ever seen stomata on flower? Anybody in the class? Do you know? If flower petals would have, everybody knows those words, right? You know flower, you know petals, and you know stomata. And the question is, do flower petals have stomata?
That's yours.
Okay, can I see more? This is fun. Huh? This one? Let me try. So one mistake here, I really want you to put it here, because otherwise you won't have the XY control. It really needs to go in this pocket.
I help.
And then you want to make sure when you focus. Ooh. So there's something very special about this particular plant that I can say, because from 2015, I have a Foldscope post about this particular flower. I think this is called the silver buttercup. And it's a silver buttercup for a very interesting reason. Do you have the macro flower now or no? It's in the petal.
That's not mine though, but.
What's super cool about this object is actually it makes a mirror. So when you're walking at the right angle, this thing will shine. And there's really a particular structure in the plant petals that builds a reflective mirror. But you need the full object to take a look at it to see how it shines. And kind of one of the threads is that by just doing microscopy, you can actually figure out. And then you might ask, oh, why would flowers, anybody online, why would flower want to shine like a mirror?
Reflect the light.
Reflect light, yes, but why would it want to reflect light? Why would a flower want to reflect a ton of light?
Maybe in some hot regions, it wouldn't want to lose out too much heat or water.
So that's an interesting idea that, oh, it could be a thermal mechanism. It could be an attention because flowers, all kinds of coloration patterns are dependent. So suddenly what we have gone from is, oh, this is the purple one. Let's give pseudonames to them so that usually we should have marked them. Let me see what this is. Is this the hard one? This is tough.
Maybe.
Okay.
I don't know where mine went. Did anyone see the one that looks like this?
Yeah, draw and share. That's actually pretty good. Okay, so first of all, it is a liquid sample.
Did you
I
see
can
it?
definitely see sand grains.
Sand?
I think that looks like sand
Thank you.
grains. Okay, so then we mount. Oh no.
And this is that yours?
Yeah.
I think.
Okay, this is quite puzzling. Okay, so I'm going to show you something in this just of how to take pictures per se as well. The point of this exercise was you can immediately realize how quickly you can go into the unknown in terms of just absolutely what you would have thought about. So let's walk through for a second. What did people see? Don't tell anybody what you collected, but from other people's samples, did you see something that you can start briefly talking about, for example? Like, I know you saw the sample that you had made. What did you see in that? What did you observe? Huh? Yes. So, yeah. And so what can you say about it? I think the idea is, what do you remember from what you see?
Like like the protrusions of the like head and versus the
Uh-huh.
tail end was
Yeah.
so clear with the microscope. It was really interesting
Yeah,
to see.
that. And what else jumps out? Actually, let me just show you all now the samples all together. So now what I've done is I've mounted this little washer on my phone on one of the cameras. I'm gonna open it up. And can I see that sample? Let's return samples back home. So whosoever has the insect, this was your sample. And I'm gonna ask one at a time. And what we will do is quickly also show people online. So if you pass that to me, I'll find it. Yeah. Yeah, it's there, as long as it's there. Okay, let me actually mount. So I'm now mounting a light source because I just want to show people online for a second what it looks like. So this is a little LED, especially when if you're working in an evening or it's dark. If you don't have a lamp or something, you can use this, but usually it's much better to actually just use a lamp. And I'm mounting now another magnet. All of this instruction is online, so I'm just going a little bit fast. So now if I've done that, then now you see I don't need any other light source. Then you can add a little diffuser here. And so this was the sample. No, this was the sample that you had given. No, I passed. This is the insect, right?
Yeah.
Yeah.
There should be
Yeah.
like a petal.
I find it. There, maybe. Okay. So
I'm not sure if it
yeah, no, it's actually, it was alive when I looked at it because I could see even the legs were beating, and there it is. And there's something very, very special in right here. Okay, so first of all, now everybody can experience the same thing that you were experiencing because now I'm doing this on the phone. I bring this close to the object now and it clicks. You see, so it's clicked now. It's magnetically. So that's the object that we are looking at now. So everybody can now see the object. And there's something very special I want to show you guys because now, of course, I also have the light source. So then I can put this down. And if you guys come here for a second, let's all huddle together. So I want to show you some advanced tricks. So I'm now keeping it down. Say I want to do a time lapse. Maybe this was an embryo that's growing. It's a little bit blurry. So I'm going to now, you see what I did right there. So now I am looking at the hair of this, and I'm recording this. And what do you all notice? So first of all, notice the hair, but you see every single little side hair. This is the wing, by the way, right? The wing is very different. It's very distinct compared to any wing that you might have seen. It looks like an insect. That's the head. That's the compound eye, the antennas. But the wing is quite strange. And you said you found it on a flower, right?
Found it on, yeah, this one. I actually meant to grab this. But
Yeah.
When I came back from the class, I saw
And
there's a
that's the
pretty-
surprise. So I don't know if you guys can see this, but now we are transitioning from observing to asking questions. That's the point of this object. The point of the object and the experience is to learn how to ask questions. Because we all observe and they are intuitively then. So when you look at this, what comes to your mind? And there's something very, very special about this object. Like, it doesn't look like a traditional wing, kind of looks like a stick, right? I mean, the dimensions of this wing are just so thin compared to, so that could be one is like, what is going on? Does it, when it fly, it expand? Now, the other puzzle is, this is a microscopic flying insect. Because the dimensions that you're looking at are less than 200 microns. So it's an insect that has the brain, has everything, but it is actually smaller than a single cell that I have in here. I have a 5 millimeter cell in here, and I have an insect that's around 300 microns. So it's super special to then say, wait a second, yes.
Does the wing look weird because at that size, air moves
Exactly, exactly. So there's a famous theorem in low Reynolds number hydrodynamics that drag of an object for this scale, you can just have sticks. So small objects could just have sticks. They don't need a wing. A stick has the same drag, and effectively that is sufficient drag. So these are called thrips. They live in flowers, they feed. Now you could, so now I'm going in, I'm connecting it to some knowledge that I have from the past, something I know about physics, and you're all kind of circling around, but it is all entitled with that observation that started. And you can take that path any direction. And we could do this for every single sample. That's kind of the point of it. And you develop sort of this repository of knowledge, but that is anchored in experiences. That's the difference. So the whole reason we started this from an educational point of view is nature is a book, but you need to learn to read it. The language of nature is very different. It's patterns, it's kind of mathematics, it's physics, it's biology, but it is not written in letters. When you read it in letters, that's somebody else's ideas, a layer that we impose. And you want to learn how to impose your own layer or your own filter. You might be excited about the ecology of this object. You might be excited about, so just so that people have seen this insect, it's quite a beautiful insect. And now when I use that word, and Feynman said it very well one time, that his dad was talking to him about birding. You all know the story. And he's like, oh, that is that bird. And in every language, his dad could tell the name of that bird. In Japanese, it's called this. Chinese, it's called this. In Indian, Hindi, it's called this. In Arabic, it's called this. And then after naming 15 names, he said, and you have not learned anything about the bird. If you want to learn about the bird, just go watch the bird. And that's the heart of science in many ways. That's kind of the heart of sharing experiences in science. And so it's not, your entry point doesn't need to be that you need a ton of knowledge, but the entry point is that you have this capacity to observe. So one thing we're going to do is now step 2 on this is learn how to collect data. And then I'm going to show you kind of the community site. So this, everything I was saying, the entire video, all our oohs and ahs, everything just got recorded. And I'm going to put this on the community site for anybody else to see and comment. And the purpose of that is it's authentic experiences. We're not pretending like, oh, I'm some big shot scientist and let me tell you what happened after all my over-analysis. I'm just sharing what I saw. And then you're just sharing what you saw. It's not about being right or wrong. So it's another medium in terms of how we express ourselves. So let's do this. Let's make sure that everybody knows how to collect this high quality data. So try this experiment yourself. You all have these little light modules now. And then if you want to start playing and you love optics, start messing around with your light module. For example, what you can do is I take a little piece of paper and I snuck it on top of this. Suddenly you see the light looks different. Do you know what this is called? In Frugal science and paper, what have I done? So I took light and I stuck a piece of paper, and you see when I let go of the paper, the light looks like this. And when I put the paper, so why would you think I would suggest you to put the paper? What is it doing to the light? A piece of paper
Dimming the light,
yeah,
dimming the light, reducing the light.
dimming the light, that's a good idea. It's changing
Opacity.
intensity because intensity is important. What else? It's doing something very important that's optically really, really clever.
Polarizing the light.
It could be polarizing. This one doesn't polarize it, but you're on the right track.
Diffusing light.
Diffusing the light, exactly. It's a diffuser. And what that does, because if I was using a point source, I would need to be in the back focal plane so I get a flat field of view. Now it's not that, and I'm using a scattering media to just scramble the light. And when I scramble the light, you'll see a massive difference. Previously, what you saw was a field of view where we had no diffuser. And now I'm gonna add the point with the diffuser. And I just want you all to see optically, what does that look like? And take a look now.
Oh, yeah.
What a dramatic difference. So I'll just show this here. You see all the colors, like true colors are starting to appear. So do you all notice that? Yeah. Massive difference. And I still have to focus. I'm just going to focus this here. It almost as if, and it's not one is better than the other. So if you guys come look here now for a second, one more time, look at that.
Wow.
Just see those structures?
Wow. Oh.
But all I did was a very, very simple thing. I just changed the illumination. There are certain things I was seeing in the past that I'm not seeing now, but there are new things that I'm seeing now that I was not seeing in the past. And so this is the notion that microscopy is about how you bring light as much as how you collect the light. So I made a very subtle change, which is 0.0001 cent, and completely changed my experience of what I might be trying to do. And again, it's just literally any piece of paper. And again, I don't need this to have that experience. That's sort of the important part of this, is that it will look exactly the same to your eye when you're playing with that. Okay, so just reminder on how I want to make sure everybody's collecting good data. So there is another little kind of a thing like this in there. There are many magnets that are stacked, and you want to find this particular magnet that has a circle. And in fact, I'm playing a trick on you guys. This accessory kit is from Foldscope 1.0, but we are using it. It's also backward compatible to 2.0. So we're using a 1.0 and a 2.0 object. I'm taking this and I'm mounting this in here with the dots mounting with the dots. You see these dots align with this dot. And there is a very important feature here. Why did we make it to be sliding? Because now we can do dark field. So I'm going to take this and off align it. So now you see only the light that leaks from the side is allowed to come. So what I have done here is this was perfectly aligned. You all see that's aligned. And now this is off align. And when it's off align, why would it give me a different contrast? Yeah, light's coming at an angle. So then when light comes through, it's transmitted light. When it comes at an angle, this light doesn't actually directly enter my camera. Only the scattering cross section enters. And that's what's called dark field. So yeah, you just want to pull that out. Yeah, yeah, that's it. Now the battery is on, and then I can show you the turning it on and off. It's always a little bit tricky because it just kind of sometimes it snaps out. That's why we have now moved to this newer LED module. Okay, I'm gonna have you hack this one. I'll open it in a second and I'll show you. So we have to just open this here and reset the battery in a second. So now if I have this on the side, what I'm going to see here is a totally different contrast, a third contrast now, which is called dark field microscopy, in which no direct light passes through. And now when you look, the background is dark. And so the sets of contrasts that come about are actually very different.
Wow.
And now I can see these tiny little dots that I was not seeing before. Now those are literally at a degree of a micron or so, because now I'm getting a contrast, not from the light that was directly passing through, but from the scattering cross-section. And scattering cross-section of an object can be larger than its own size. So with dark field, I can see 100 nanometer gold bead or a bacteria that's very small, that is below the resolution limit of your optics itself. And why is that so? How can you see something below optical resolution in dark field? The answer is hidden in scattering. You see, the problem with transmitted light microscopy is you have to absorb enough light to make a difference. But scattering cross-section is, if you crank up your light in transmitted light, the contrast around you also cranks up, right? More photons you pump, but where there is no object, there's more photons coming from there, so sensitivity doesn't change. In dark field, you can crank up your light source, a lot of photons are going through, and you can increase the number of photons you would see by reflection because the direct photons don't hit the camera or your detector or your eye. So now you are in this limit that you will get a spot, and as long as you have X number of photons that were emitting, you will not be able to tell the size of that object. That will still be the resolution limit, but you can detect an object that's below a resolution limit. So it's a very crazy idea that you could see a 10 nanometer particle if it has enough reflection contrast. And that's why we use gold and many of these other sets of beads and many of these single molecule assays, because in dark field, you can crank up the illumination. So kind of one of the tricks that is important is, that's why I want you guys to all make sure that you play and get it right. So when I collect the data, I always like to put a book or something so your phone is fixed. And then I need to show, so I'm gonna walk around and make sure that everybody, whose sample was this? That everybody has this right. And so a couple of tricks. So first of all, to improve your field of view, I remove the color. It's just because you'll get more of it. Secondly, never look at it under a cell phone unless you first get a spectacular image with your eye, right? So we're gonna make sure, and of course, no surprise, if you believe. I'm seeing the Venetian patterns. There's something really interesting. You know, the moment you break a leaf, there are all these air pockets that appear. And so I'm seeing many of those air pockets in there that I haven't actually seen before. So let's look at this junction. I can see the cellular structure. It's really, really beautiful. Now, step two is let's mount your coupler. Yeah, so let's find that. And then we should look at what that is.
Yes, I thought it was always with the cell deck.
Yeah. So when plants get infected by viruses, they do this really crazy thing. Nobody's looking right now. They kill themselves. So this is like a burn zone. So there's possibly a viral infection on the leaf. Exactly. They are killing, it's called autophagy right there. So do you notice that yellow zone, the dark is all dead? And this is the only way that the plant can actually survive because it's not going to spread completely. Okay, so you peel this off. And the first thing with some phones you have to figure out often is which one is the camera. So I like doing video mode. And in video mode, I zoom in a little bit because I want some digital magnification, and then I block the cameras to check which one it is. And so at 2.1, it's the top one. Which phone is this? I've not seen this phone. 10X.
iPhone 10.
No, wait, wait, wait.
Now that, no.
All right. So now I'm recording and I'm in here, and I like the idea of just being able to hold the weight like this. First go in. Right? Now I like now, so you have all the handles in place, and now you can see focus and I'm playing and playing. I want to make sure there.
Look at those, still not.
Right there. Oh, I should. Oh, we recorded it. That's good.
Nice.
I recorded that.
And
And so now you can just back off and run a time lapse, and you would watch these things open and close. Right? So suddenly you realize, and it's very important that now you're collecting high quality data. That's why the phone is not in your hand. It needs to be sitting down. Even when I'm outside, I'll make a block of stones to make sure. If I'm, I mean, you know, the expectation is you start with exploration, but at the end, you want to really be pushing your limits of what you can see. And so now once this has happened, I like this as a way to move around. So I go here. This is what I was saying, that all these little air pockets that have appeared as well. And if I want to do a quick Z, if it's a 3D object, you see what I'm doing? I am letting the weight. So sometimes when it's really dynamic, it's moving around, I don't have the time to get to the Z axis. I use weight of the phone as a way to do quick Z scans. Now I wish we had brought some coral today. With the terrestrial world. And then one important aspect about this is there are many modalities that exist. So for example, for time lapses, I use an app called Lapsit, and it gives me exactly the exposure times, and you start really controlling just like all the other parameters that you would want to control. Okay, so make sure you guys have a high res. Actually, don't move it for a second. I just want to show the class. Just a 10-second. Oh, that's a cool sample. While we were talking, a spider has shown up. Okay, give me a card, quick. Anybody? Can you all see the spider on my face? The spider really wants to be inside the Foldscope. I need a tape also on it so that, yeah, and we're gonna try to look at the spinner rate. I don't know how many of you have seen how spiders generate silk from their butt. Oh, you gotta do it quick. Okay, anybody has a tape?
Everyone try to get a tape.
No, no, I'm getting to.
Here.
Here.
You're good at it.
Come on, come on, come on. Mount it on there. Mount it there. Okay, I can't do this anymore. You know, it's eating its spin rate so quickly. And
Is he eating it?
okay, I need another tape. Quick, quick, quick. Tape number 2. This is what happens with. Ah, we got it. Okay. Tiny little spider, and it really, I didn't ask for it to join the class, but it came and it hung on the camera, and let's choose that.
Good.
And we're gonna mount, unless you were imaging.
No, it's fine.
Okay, let's try it. Oh, and then by the way, the cards that you are making like this should look, I will show you my personal card, and this part is important. This is really designed to create a global trading card game. And so what I often will do, I'll take the card. And I have around, I was counting, roughly around 1,000 of them from all kinds of places around the world. And so this is one example. So this is a very special card to me. Suddenly it's very personal because it has a very, very special sample. And so I've written it, I've signed it, I drew it, and I wrote the exact GPS coordinate systems of where I was. I know I collected the sample from a kilometer deep in the Pacific Ocean. And I was really surprised to see something that I know was first described in 1850. So suddenly, these sets of objects, this spider might be, the most common spider in the world, but it might be something that hasn't been described. And I don't know. And the purpose of this is so what will matter is if once you have made this card, you really write down, and then it's of personal importance to me. I don't care where it will stand in science, what will we learn from it later on, but at that moment, it taught me something. So it's very important. You want to bring your artistic side to the table and you want to write your observations, because this physical object we want to leave for the future. And the new program that we're just starting with a sequencing company is actually to then upvote in the community. So if I was to say one sample from today, that's the most awesome defined by people, will be sequenced. So which one should that be? And so that comes from this. And so this archival information is very important. And currently, in much of what we know in microbiology and microscopic world is not geotagged to any location. So this spider is not just about the fact that it showed up. It matters that it was here. This is a completely missing information. So if I give you an organism name, nobody would know where to find them in the world. In fact, we don't even know what's the rate of species loss at a microbial scale. Like that thrip, is that the most rare thrip that has ever been seen? Or it's something that association has never been seen? We will never know. Because most of the time, even when we describe species, we strip them from any ecological context. And when something is described, we still don't know. So it's very important that you write down and make these cards as an archival object. Okay, so that's why I wanted to make sure whosoever this is, that person finds their sample back. But I'll do a demo just to make sure that everybody is, and if you're having a little bit of trouble getting the data correctly, then talk to people around, practice it now, because then you won't be using otherwise your Foldscope to the fullest. So one important thing that I also did in this sample is I did not squeeze it. I was very gentle with the two tapes. So my intuition is most likely the spider is alive. We'll find out. And my intuition also is that, so let me ask you a question. Do spiders have hearts?
Yes.
Yes? Where do you think their heart is? I'm going to use a little piece of tape.
In their
Right or left side? It's a trick question.
back. It's in the back.
Yeah, yeah. So people online, do spiders have heart? And if they have a heart, is it on the right side or the left side? Trick question. Yes, they do have one on each side. Yes. You should first ask, do they have a circulatory system? Yeah, they have an open system. So that changes lots of things in terms of how you might even ask that question. And you can be completely naive about it and just go and look. That's kind of the point of it, that you don't have to have any sets of scenarios, and I could just pose a question. Okay, so I did not squish it. It's unhappy. And I really like the sample. I'm trying to see. Oh, there is a piece of tape right here. This is what I was looking for. So when you will run out of that little special piece of tape with the little thing, this is the solution, just regular Scotch tape that's clear, not the one that has, the diffusion on it. This is scissors you're carrying, and I'm gonna mount this on top of this as a diffuser. So I mounted that, I mount this, and then if you guys come take a look, let's just do a little bit of spider biology here. So if you guys just want to come for 1 second. So again, a couple of tricks. I'm doing this quickly I always start with video. I always start with a little bit of zoom. And since nowadays there are so many cameras, I check which camera should it be on, right? So it's not on this. So sometimes you'll be like, why can't I get the image? Because when you zoom, they flip to which camera that they would go. So I'm here and I often record a little bit before I start imaging. It's just so I know the context. We just caught a spider. It was hanging out on the screen, Kevin's screen in Zoom. It was trying to join the Zoom. We caught it and we're going to take a look at it. And so then when I just bring that in, it's snapped in. It's sitting there. And because I don't want too much weight on it, I'm going to make sure. Can you guys see it already? So those are the legs. And something really funny. It looks like as if it's a stump. Yeah. Which doesn't make sense, but that's because the spider's folded its legs. And that's the spinneret. You guys see? Right at the tip. Oh, there. Oh, and the spinneret is moving. Did you see it move a second ago? It's contracting. There is a valve there, and that valve is how it holds to the spinneret. So it will send the silk, but it needs to hold to the silk as well, because otherwise it'll still flow. Wow. So, and I'm going to change the contrast in a second, but what I'm very happy is that I don't see any oozing fluid. That means it's still alive. The legs are folded because they're taped together. And let's get to the mouth first and the head. Oh my God.
Manu, we are having FOMO.
Oh, there is the heart. Okay, let's have them show. Kevin, can you pick that up? They also want to see. Okay, right there. So first of all, immediately you notice there are little, right there. Whoa, wow. Do you see that? There's a little pump inside the head that was beating a second ago. And you also notice that there are holes. Oh, whoa. Do you see that? And do you see the holes in the back? There are little holes. Mm-hmm. Yeah. Why should there be holes in the back? Like the body has as if bullet holes are there. Exactly, gas exchange, because they don't breathe like we do. So these are spiracles, because most insects and spiders have these spiracles to do just diffusion. Now you might ask, oh, this is another folded leg. You might ask, oh, what is actually inside the leg? So I'm going to focus on a leg for a second, right there. And if I zoom, do you see that? That's the immune system. Did you guys see it for a second? That's the hemolymph has immune cells, and you're seeing them just zipping by. So fast. Yeah, just so we know it has a heart because it's pumping. Do you guys see these little cells zipping by? And I'll post these videos online. So suddenly you realize, oh, wait a second. It has the same kind, but it doesn't have blood cells. These are larger macrophages. So if there was an injury, and what I'm really surprised by is the speed. So I've seen it in other insects, but never caught it live in a spider. But the speed is insane. I mean, yeah, like literally it's zipping past. So did you get a chance to see? And yeah, I mean, now you could just turn your phone and do the high-speed imaging on the same. So I could go in here and convert that, but high-speed changes resolution. So now what we want to do is we've collected a little bit of phenomena, and I could just spend like 2 or 3 hours just observing sets of things and writing observation. And the point is, you arrive at this object with just open mind. You just are there to see. We have no bias. There is no agenda today. We didn't plan to do a spider biology lesson or anything. You just want to put pieces of observation together in your mind. And that's kind of the purpose of this object. The object is designed to just share experiences of the living world with anybody and everybody. And one of the things that happens is the more you do it, the more you can interpret. So I knew that I need to be very sensitive to not squash it, because otherwise all of this would have looked like a blob. You take a picture of it, you take a photograph of it, none of it's there. This is sort of very important to understand. Microscopy done in a context with physiology is sort of what starts teaching you lessons. Now, for example, if I am here, I could have been in a cold place. Maybe I caught a spider when it was very cold outside. You would explicitly see what the role of temperature might look like. You might see one set of a spider that has this flow rate, and then I could now take in, actually, I'll ask you a question. Because we saw a heart pumping, what if I added a drop of caffeine in this slide? What do you all think? Any show of hands? What do you think would happen?
Faster.
Beat faster. How many people
Does
say
caffeine
beat
work on-
faster?
I think it would kill.
We have some.
Okay.
It would work. I bet it would.
Some
I bet
people,
it
okay,
would drown.
how people think it'll kill the spider, beat faster. What else? Anybody else has another idea? What would caffeine do? And you could just say nothing would happen then, right? Okay, it'll panic. Yeah. So when you do that for certain class of things, and this is actually a very classic experiments because scientists drink coffee and they're doing microscopy, you always add caffeine. This is not a made up thing. Caffeine is a very classic drug perturbation assay. And when I did that last time, not for a spider, but for a Daphnia, it went into a heart arrest, arrhythmia. So it was pulsing perfectly normally. I collected for 20 minutes and then suddenly it became erratic. And that's because I didn't calculate the concentration. Think about that concentration in the blood for what we are thinking about. I didn't dial in. So any of our intuition about heart beating faster is all a function. And so then suddenly, if you think about pharmacology, I went from spider drinking coffee to pharmacology. It's like, oh, it really matters that drug dosage needs to be known as a function of physiology of the organism. So I'm just sharing these stories because I want to tell you that this is a random path. And so when you start doing this, you will feel like, I don't know where I'm going. I don't have an idea, but you're not supposed to have an idea. All of this is post-reflection. And when you observe at that time, you have even no clue what you've just seen. But it's important to do it over and over again to then make these reflections. And then you say, oh, now I understand how to process those ideas. I have enough tools in my toolbox to process this. So it's, for most people, microscopes in their life have been, oh, we saw it. Yes, I know what a microscope is, right? For majority of people in the world, first of all, majority of people have never seen a microscope. Then the ones that have seen are like, oh yeah, I saw an onion cell. I know what a cell is because they have this preconceived notion that cells are like bricks or what Robert Hooke had described, or cell is not like that. Cell is anything you can imagine and more. And so in some sense, we start with these preconceived notions, and the purpose of this is you strip that away and you just spend time observing. So one of the things that is important about, I gave that analogy of a bicycle. You only get good at biking if you do it every day. You take it to mountains, then you start jumping off of airplanes in your bike. Like that's the purpose. So I was someplace and I wanted to know what tree canopies look like in some rainforest in Costa Rica. So we mounted this because you can see this is now a fully autonomous unit. I could be in a tree canopy doing this. It has a battery. It has a cell phone. It has charge. I can connect to it. I can do live stream. So as an instrument, this object is very powerful. So you could be in a volcano and doing microscopy. Or, at that time, I was in a tree canopy looking at this ant species that lives inside the flower of this orchid. And it's only found in these very high heights. Those ants never come down. So the purpose of all of this is also try taking your Foldscope to new places. Because you would conventionally not. So you would be in the middle of a boat. You would be in the middle of a grocery store. You would be in the middle of a coffee shop. And that's kind of the social contract around this. The social contract around this is that when you do this in a coffee shop rather than a lab, a whole bunch of people will be like, what the hell are you doing? And if they don't kick you out, that I can tell you sometimes does happen. You have to handle it carefully. But if they don't kick you out, the next thing, no, they won't kick you out. I'm just joking. Then you start conversations. And kind of this is the social contracts around education. Is that if you're thinking about science education, it is not for a particular group of people, it's for everybody. And you have to learn that practice. So we call this street science. And there was a Foldscoper from Iraq, Muhammad. He wrote to me, I would say, during the Iraq War. And now this is the last story, and then we'll end. He wrote me like a 2-line message saying, Manu, I'm writing from Kirkuk. I would like to have some Foldscopes because I want to share that with kids. Actually, this was right before the big war had started. Or it was not in Kirkuk at that time. And I was like, yes, you know, we don't have a community in Iraq. I'm just going to send them out. We figured out ways to get them there. It was hard. And then eventually, I didn't hear back from him. It was a full-blown war. Nobody knew what was going on. And I think around 6 years later, he writes back an apologetic message that, Manu, I'm sorry I've been out of touch. I could not do what I had promised. I apologize. I had no mention of war, nothing. And I was like, no, Mohammed, I'm happy to hear that you're okay. And I know about what's going on. And he's like, no, I didn't fulfill my promise, but now things are better. I'm going to now start sharing. So we developed a friendship and he started going out. And then I started this challenge. I called it the Street Science Challenge, which is make an observation, a new observation every day. Like even within the last hour or so, we have seen 6, 7 completely new things that I had never seen. I've tried to watch the spinneret for many years. And every time either the spider is too squish because the pressure changes. And I've never seen the spinneret valve before. So we already saw a new thing. And so I started doing this and I continued it for 30 days. And I wrote my notes. Every day at 9 o'clock, I'll start 9 to 11. I will make observations and they have to be new observations. So this, I'm just throwing it as a challenge to you. If you really want to be a Olympic mountain biker, this is what you have to do. You have to observe every single day at a kind of an intensity that nobody has seen before. Now, when you do that, at 30 days, I was like, okay, this is good. I compiled it. And then I shared that in the community. And then a couple of years passed and then I saw Muhammad for around, his record now is around 145 days, continuously running workshops and observing at night in streets of Iraq. So he's not just doing it alone, he's actually doing it with kids. And every day he would write this blog about this is what happened. Many of the blogs are about science and what he saw, but many of them are about violence. Many of them are about broken trust. Like because Shia Sunni was a big issue and he belongs to one sect, he would walk into a region, the first thing people would be like, oh, what sect are you? Or like one time he was chased away while he was working with kids at a bicycle shop because somebody didn't trust. I mean, of course, it's a war zone. Many of these kids haven't been to school for a long time. So it suddenly starts opening and unraveling and thinking about that what you're trying to do is in a context of a society. And that society has its own sets of reflections. And you're not trying to do it in isolation. So, I mean, it's not about, oh, you can't politicize science or you can't. It's all in one because it's happening in the same space. And so it's very valuable that I'm throwing this as a challenge for you guys that, of course, you will learn a lot from this object. And that's why everybody gets at least 2. But howsoever many there are, because some people didn't show up today, you guys can have as many as you. No, we'll give it to them too. But the idea is now share this with someone else. Like share this authentic experience with someone else. Kind of bring them up to a level where they feel excited about sharing that in a community. Or take it to a path that they might want to take. And so that's kind of the most important aspect of this, which is the social or now, up till now, it was all about science, but this is far more about science activism. And especially for some of you who are thinking about very specifically solving problems in education. Because at the end of the day, it's something that you will only be able to reach X number of people. But if you don't build things that has a self-perpetuating loop or like a PCR reaction, it's not going to spread. And so this is kind of the whole point about people joining right now. It's very important that the kind of experiences that we build and the kind of tools that we build, you can take the same philosophy that we built for Foldscope, but actually apply it to a totally different tool. You can apply it in education, you can apply it in health. It's not a function of that, oh, this is a replication. It's just much more of a model system. So that's kind of what I want to say. We'll do an open mic. I know there was a question. And then can you remind me your name again? Raina. Yes, go ahead. Yes. Yes.
Also just thinking about it, like Iraq's also had a lot of bombings, violence, things like that. And so nature naturally would probably contain some remnants of those kind of
Yes,
things.
yes.
So like, was that
Yeah.
ever captured?
Can I tell you a very sad story? One of the Foldscope posts I read from one of the kids was actually trying to figure out it's called bullet forensics. And he posted a story about how he was able to decipher which gun, this particular gun, and from the gun you can tell who owns it, a particular bullet was fired. Because when a bullet gets fired, it doesn't just go straight, it actually rotates. Every gun has a very particular frequency. So if you ever find a bullet, you take a tape, you stick it, the number of lines and the pitch of that helix, you can actually go back and decipher particularly the weapon. So, I mean, that's the reality. And I mean, in some sense, it was like, whoa, how did this kid somewhere in the middle figure all of this out? But on the other hand, you can see the intent. What he was trying to do was figure out who fired this bullet. So, and I mean, it's stuff like this is everywhere. When you talk about, I mean, many of the things and places, we have communities in the Amazon, they are about gold mining. Many of the posts are really about kind of what's happening from an ecological point of view. But at that same time, if you dive deeper, you kind of realize the way illegal gold mining has been going, it's actually a kind of slavery, because generations of people are required. The reason they remain in forests is because they're not allowed to go. And because they are told that your father had a debt of this much, so you're going to pay for it. And then something happens to the parents, and then suddenly now all of these people are just working. And then now 30 years have passed, they have no idea, they still continue to do the same thing. And so when you interact with people, sort of reality spills out in ways that goes far beyond science. And then you might say, oh, I'm thinking about a solution of a problem in illegal gold mining. But if you don't understand the slavery that's in play, you'll never be able to. It's not the person who's actually digging and doing the dirty work that's responsible in many ways. So I think this is the rawness of much of interacting kind of as humans with each other. And it's important, it should come out. This is why the community site doesn't have a template. We're not asking people to write papers. It's just a blank sheet of paper. You do what you want to do. I mean, another much more positive example is 2-3 years ago, there was a massive set of projects from Mexico around pregnancy pills and pregnancy tests. And kind of one of the threads that was associated is in many sets of countries, just the act of buying a pregnancy test marks you like, oh, something is going on. And that's very taboo. And so suddenly there was these sets of posts of thinking about, oh, how can you actually just do private tests without having ever to buy anything? And in the old days, like before somebody, did I tell you the story of peeing on the frog? What? Okay. So pregnancy test came because somebody accidentally peed on the frog and the frog started ovulating. That's how we discovered that there was actually a hormone. And this is kind of the bizarre world of cat whiskers, and science is built with things like these. That pee contained the hormone that needed to be detected. And for a long period of time, it was completely illegal to find out whether you're pregnant or not with religion and all of these sets of things. There were underground entities and women groups that would ship a live frog if you wanted it. And that's kind of the reality. And then somebody says, I think we should stop shipping frogs. Let's actually figure out what is the interacting enzyme. And that's when they deciphered the sets of hormones that are now detected as an RDT. So this is a total sidetrack. What I wanted to say was even before the peeing on the frog, there was actually a thing that you could do that the saliva, the way it dries, actually changes. So crystallization patterns in saliva, crystallization patterns in a tear changes, and there are correlations. It's not perfect. And so kind of one of the threads was, oh, could we revive? Could we understand how a teardrop crystallizes because there are hormones and compounds in it and salt concentrations in it that could be deciphered to then build a stealth pregnancy test for anybody that they can just build at home. And that was kind of the project from a group in Mexico. And, you know, it's still open. We did a lot of different sets of things, but I'm just trying to say this, that humanity is integrated in ways you never understand what the real problem is unless people have a chance to express. And then when it's coming from an authentic place. So it's very important that if you're sharing with this somebody, like, it's not about us scientists getting to at somebody's level. That is just total bullshit. We're all humans. We all have same kinds of emotions and sets of scenarios, but we just have to connect. When you're connecting with people, connect at a human level. So nobody has more knowledge, less knowledge, because they might know more about something than you do. So it is an art. It takes a lot of time, and it sort of has to be done with intent. It's not something that you walk into a place and you're like, oh, let me tell you. That just never works. And it also has to be done with practice. The first time it'll totally bomb. You'll be at a bar, you think you got this busy bartender, you want to show them what's in the martini, and he's like, I don't care. But that's just, this is an art. Like, I think the best analogy I can give you, Foldscoping is like improv comedy. That's the most thing, like improv comedy, true improv comedy, like when Robin Williams would stand and talk. I don't know how much of that content was written and how much he was just blurting out. Like, if you know your art so well, so science communication must really feel like improv comedy. And I mean, even that word doesn't make any sense because it's just a natural thing that you do, but you have to make sure that you practice it. That's what I meant. Like, people go to thing, they stand in front of people. You're vulnerable. When you're going to stand in front of 30 kids, it's like, oh, this guy knows about plants. They're going to throw all kinds of stuff at you. Not literally, but figuratively, sometimes literally. You just have to be ready for it. You have to understand that, oh, I've set the baseline that not everybody knows everything. So kind of, I think I'm kind of now bleeding into this notion of just science education in general. It's very important for our society, especially with what's happening currently, like everything is going to shit. And it's very important that we engage, but it's also not, it's not a passive. Again, it's a contact sport. Okay, open mic. I'll shut up. Any other comments? Take everything that's on the table other than laptops. Anybody wants to take?
What do people bring?
Oh, yeah. So let's figure out. So that was a trip. We figured that out. I could not figure out Enrico's sample, so I cannot take that. I am really curious about that sample. So I want to look at that more. One guess I have, which might sound crazy, but for a second, I thought it was boogers. No? Okay, hope not. Anyway, they kind of look like boogers. It has the mucus, it has all the kinds of things, but maybe not. Other ones, I know that was a buttercup. That was pollen and anther. A dead leaf.
Spider silk.
Oh, I want to see it.
Really cool.
I want to see it. This I did not get a
That
chance.
was that's why I walked in like this. People thought I was crazy when I walked back home. Like,
Because, oh, this has.
sorry.
Yeah.
I put a little slide in
That's
this.
good. Yes. Okay, any questions? Open mic. Anybody wants to talk? Raise your hand and you can talk. No? Okay, so then we'll say bye. All of you, please send your address to Kevin for shipping more Foldscopes. If we are shipping far, the farther you are by distance in miles, I'm going to multiply a factor, the more Foldscopes you will receive. Do you understand that? The farther you are in distance from Stanford, the more Foldscopes you will receive. So if you're on Moon, you will receive 1 million Foldscopes, and we will scale accordingly. Shit, I might have, did I do this right? Okay, we cannot do below 1. Yes, we'll figure that part out. But anyway, the farther you are, because it costs more money. Oh, she's going to the South Pole. You could actually figure out, that's true. If you give me an address that's farthest from Stanford, then you get more Foldscopes. Yeah, the point is just shipping costs. It cost us more to ship you 1 Foldscope and in that same amount of time, but there is also a responsibility. If we ship you more Foldscope, we expect that you'll share that with other people. And oh my goodness, holy crap. Individual fibers are truly visible. So I think what's going on, this is a species. So spinnerets, when they spin, It's actually multiple spinnerets.
Hmm.
And for some species, all of them join. But I think the species that you have, the spinnerets, they are so small
Yeah.
that it's possible that they don't. Because silk is made for many different reasons. It's not just for the web, it's made for cocoon, it's made for putting eggs inside. And so it's a choice that the spinneret could make. I'm just going to show you a sample that Danny has just put. Just like 2 minutes. Hey, feedback. Yes, you guys, you can talk to them. I'll leave in a second. But we're having too much fun right now, so you have to wait. Okay, so I'm just gonna show you guys what Danny just found. And it's exciting for many, many reasons. And the biggest one is, ooh, there's a second phenomena that you see, that some of the silk fibers are decorated with something. And remember, I had said that the pollen was charged. So it turns out people have discovered there is electrostatic charge on the web. And it's possible that dust and many other particles are actually, do you see that?
Mm-hmm.
These are charge interactions. So
Wow.
sorry, just gonna show that here. Do you all see that? Oh, that was coffee.
Yeah.
Oh, that makes sense.
Yeah.
And but okay, it had milk in it.
Yes.
Yeah, so that's the lipid droplets.
Okay.
Yeah, that's what I was thinking, that it's the, because coffee you would see the actual grinds.
Yeah.
I should have won that one. Okay, so make sure that all of you really document your cards, and then let's see how many when we meet next. Oh, this is incredible. So what's really cool is inside the spider silk, a trichome. This guy would know what a trichome is. These are single plant cells on the surface of the leaf of a plant that fly off. They make the chemistry. You see like a Medusa-like structure, and it's stuck. So there's a single trichome that's landed on the spider silk. You see a hair-like things as a bunch, and all the chemistry that happens on a plant actually happens inside. Most of it happens in trichome. So when you smell, it's that. When it's minty, it's one of these. And these are the beautiful model system for developmental biology, because you might ask, how do you make branching structures in plant cells? So this is really, really funny. And then because they're small, they float around. Okay, so try drawing what you see. I'm gonna run, you have all the Foldscopes here. So if you don't have some particular parts, I'm gonna say bye. And if you guys wanna catch me about projects, there was a discussion around Friday, some people can't come. We will do some presentations on Friday that people can, but majority of them will be on Monday. So let's just choose Monday, actually. Let's do a demo on Friday.
Okay.
Yeah, let's do Monday as mid-project presentations, which is Fred Park. Do you have a big question and a big idea you want to work
I
on?
was so confused, I couldn't find it.
And then to save this tape, I just do that. But then when this stickiness runs out, you take a regular piece of tape and you stick that around to find on your phone. Okay? All right, I'm gonna run and I'll let you guys fill out form. These